All right, this is just a relatively short screencast to talk about the first part of the skin cancer case study. Just in case anybody's not in class when we do it, or if they miss the review session, want everybody to know this. So the first question is just explain how cells use DNA to store information and what that information is used for. And the answer to it is relatively straightforward, right? The information is stored in the base pairs, the A, C, A, G, C. It is the order of the nitrogenous bases in the DNA that encodes your genetic information. What that code does is call for, or code for, a certain series of amino acids to be strung together to make proteins. And then those proteins are basically the machinery that make your cells work. So you have transcription followed by translation. I don't go over transcription in this because I'm assuming you remember. Let me go back. It is just starting with DNA and then making a single-stranded copy of RNA called messenger RNA that is going to be complementary to the template strand, or the strand that is being used to make this mRNA. So it's got the same information that's in the DNA except U's substituted for T's. And then you will recall in the cytoplasm, the ribosome reads the mRNA and the tRNA delivers amino acids to the growing polypeptide chain. So wherever there is the three-letter, or four, I should say, every three-letter codon, there is a three-letter anticodon, which is always matched to the same amino acid. So every tRNA, every tRNA that has this anticodon is going to have that amino acid. Every tRNA with this anticodon will have that amino acid. So they're always matched the same anticodon to the same amino acid, so that anytime the same codon, like U here, shows up at the ribosome, the same tRNA with the same anticodon complementary to that codon is going to show up and deliver the same amino acid every time. And this is just the same pictures from your book. So if you look here, right, this is your mRNA, ACA. The anticodon that is complementary to that is UGU. So this tRNA sticks here because these bases want to stick to each other. They will hydrogen bond with each other. And that is what's delivering threonine, I believe is that amino acid. And then this tRNA with its anticodon is always going to deliver tryptophan. And this growing polypeptide chain will be attached to the end of tryptophan. Right, so here's how the code works. You have your template strand in your DNA, which is used to make your mRNA, and then your mRNA codes for different amino acids. So if you change one of your bases, you're going to change the codon in the RNA, which will then mean a different anticodon will be complementary to it, and maybe instead of methionine you get proline, or instead of, well, I don't know which one that's supposed to be, maybe glycine or guanine, you would get serine. I think that's guanine because glycine is GLN. I don't know. So that's the long and short of it. And right, just to review this part, right, every time your cells go through cell division, they replicate their DNA. So they pull apart the original chromosome and then build a new half onto each of the two original halves, and then you end up with two new chromosomes. This represents an opportunity for mutations to happen. And there might already be damage or mutation that's happened if this DNA has been hit by UV light. And if it's not replicated properly, or if UV light damage isn't fixed before replication, or fixed improperly, you can get mutations in the DNA or the wrong bases, which lead to the wrong amino acids, and then the protein has the wrong structure, so it's going to have the wrong function. So I asked you about two different classes of genes associated with cancer. One I should have called protooncogenes. So these, as it says down here, encode proteins that promote cell division. These proteins are generally always present, but not always activated. So it's much more efficient time-wise to have all of your proteins already synthesized, and then when it's time for them all to jump into action, you just enzymatically modify them all, and that makes them active, and then you get cell division. So we see that a lot. It's a lot easier to activate existing proteins, or a lot faster, than it is to synthesize new proteins when they're needed. So this is just one example here, but a very common example of some oncogenes. This right here, EGF, I believe stands for epidermal growth factor receptor. So it's just one receptor on the cell that can respond to signals that cause the cell to divide. And these proteins right here are going to respond to the presence of the growth factor on the outside of the cell, and they are going to cause DNA replication in the nucleus. These exist in an on form and an off form. So there's a specific part of them that gets modified to make them active, and then modified again to make them inactive. Your proto-oncogenes can generate or can have what is called a gain of function mutation. Then they are called oncogenes. So in this case, the protein is mutated in such a way that it is in its active form all the time. It can't shut off. So it's always doing its part to encourage the cell to go through mitosis. So this might, as it says here, just lead to an increase in the rate of cell division. And if you're dividing faster, you might be generating more mutations. Or again, it might just be a cell that shouldn't be dividing at all, but continues to divide because it's no longer waiting for the signal. So in a normal cell population, you might get no cell division in the absence of this growth factor. If you mutate one of the genes for these proteins here, and then that protein is always active, you're going to get cell division in the absence of any stimulatory growth factors. We tend to think of the proto-oncogenes when they become oncogenes acting right about here, right? So these are the biochemical signaling pathways or the proteins that are involved in, oh, I'm sorry, I meant here. We want to be here. This is what is initializing the entry into DNA replication or DNA synthesis in S phase. So this is what's really setting in motion the beginning of the next cell division. And there are, to be certain, other proteins along the way, the whole way through this, that continue to push the cell through the mitotic division. But this right here, this checkpoint right here at the end of G1, is the initiating part. And if that gets screwed up, it's really easy to get more cell division than you're supposed to have. Then you have tumor suppressor genes. So these genes, when they are functioning normally, prevent the cell from dividing when it's not supposed to happen, among other things. So they can be simple DNA repair and proofreading proteins. And if you look over here, this is just if you have, let's say this is a UV light caused some damage here, and you want to fix that UV damage before you go through mitosis or DNA synthesis, there's a whole number of proteins involved in removing that damaged portion of DNA and then replacing it with the proper complementary strand. So if these get screwed up, you're going to have mutations in your DNA when you go through S phase, and then those mutations are going to be passed on to the progeny of this original cell. There are also, if you look here, these two checkpoints, and even this one where it's red. Right? There are times when the cell can decide now is not a good time to divide, like if there's not enough nutrients around. But it's these two that I really wanted to point out, right? So before you enter mitosis, after you've been through S phase, you're going to check for DNA damage. And then before you complete mitosis, you're going to make sure that your chromosomes are aligned properly and that you have your mitotic spindle, because that's going to be what's responsible for making sure that the chromosomes are segregated properly. If that gets messed up, you can have massive, I don't even want to call it mutation. You can have extra chromosomes or missing chromosomes, which is a tremendous genetic insult to a cell. And then there are even proteins that can initiate what is called programmed cell death or apoptosis. And a cell can, if it is damaged enough, or if it receives a signal from the immune system to tell it to take itself apart, it will very carefully start taking itself apart on the inside so as not to burst open and cause inflammation, and then remove itself from the body before it can divide and pose a threat. These genes, these pro-apoptotic or pro-cell death genes, are still very much a hot topic in cancer research. So with tumor suppressor genes, if they get a loss of function mutation, this means that the protein does not work, and now the cell has lost one of its guards, one of its safe checks against inappropriate cell division. Or it may have lost some of its ability to repair DNA. So it's going into DNA synthesis with more DNA damage than it normally would. And so it's proofreading and checkpoint operations are going to have to be really good to catch the DNA damage. There are lots of other, what's the word I'm looking for, examples of tumor suppressor genes, but we don't really know enough about cell biology to go into all of them. And then also, right, if you mutate one of the pro-cell death genes, you can have a cell that is genetically very messed up. Maybe it has the wrong number of chromosomes, too many or too few, which can lead to all sorts of problems. Under normal circumstances, a cell like that would enter programmed cell death. It would essentially kill itself so as not to become a threat to the rest of the body. But if you mutate the important genes in these cell death pathways, then the cells don't die. And this activating cell death is one of the important functions of your immune system. So one of the things your immune system does is trick infected cells into initiating, if they're infected with a virus, I should say. It tricks virally infected cells or foreign cells into initiating programmed cell death so that they deactivate or destruct themselves. So it's a very powerful tool for maintaining homeostasis, and is the perfect kind of tool that you would want your immune system to have at its disposal if it were able to recognize cancerous cells, which sometimes your immune system can. But if the programmed cell death pathway is not functioning in that potentially cancerous cell, even if your immune system can spot it and try and stop it, it's not going to work because those genes have been mutated. All right, then. So how do we put this all together and get cancer? Now, this is what is called the multiple mutation or multiple hit theory. As far as I know, this is still basically how scientists view cancer, but I have not read up on oncology in a while. But basically, you start with a mutation or two in a tumor suppressor gene or an oncogene, and cells start dividing when they're not supposed to, or they lose some of their safeguards, so they're not being as careful as they normally would. This then increases the chances of forming more mutations. So you can get additional mutations in tumor suppressor genes or oncogenes, which would then lead to more out of control cell division. And then the more out of control your mitosis gets, the more likely you are to generate more mutations that go unchecked. And if you get mutations in genes whose proteins control certain very important cell functions, this can then change the behavior of the growing tumor or the behavior of these growing cancerous cells. So they may end up being able to take apart a basement membrane, which would then allow them to move into surrounding tissues. Sometimes the moving is just the tumor is getting bigger and it's spreading, but there are times during growth and development where cells in your body actually locomote and power their own movement. This is a normal function that tumor cells can develop, and then they become mobile. Tumors can develop the ability to stimulate blood vessel growth. This is called angiogenesis. Again, this is another something that your own body's tissues have the ability to do, right? As you grow taller, blood vessels are going to grow, or even if you're done growing taller, let's say you go to the gym and you put on 10 pounds of muscle mass, you now have more muscle that needs more blood. You are going to grow more blood vessels to bring that bigger blood supply to your now bigger muscles. Tumor cells can, if you kind of put some of these things together, generate the ability to move, take apart basement membranes. They can take apart a basement membrane, move into a surrounding tissue, maybe find a blood vessel, crawl into the blood vessel, pop out somewhere else in a distant portion of the body and start a brand new tumor. That's called metastasis. So again, all of these things, disassembling basement membranes, moving, causing blood vessel growth, crawling in and out of blood vessels, these are all things that cells in your body do, and it's part of homeostasis when it's the right cell doing it at the right time. Cancer is just when the wrong cells develop these abilities and perform them at the wrong time. And then you get large numbers of cells that don't do anything useful, invading multiple tissues or multiple organs and crowding out the cells that are trying to do their job and preventing those organs from doing their job. So if your lungs are filled with tumors aren't good at gas exchange, you're going to have trouble breathing. If your kidneys are filled with tumors aren't going to cleanse your bloodstream, and your kidneys are going to fail, and you're going to die of kidney failure. So this is one of the reasons why cancer is so difficult to treat. One is that it is derived from your own body cells, so your immune system is not super awesome at recognizing cancer because it's cells that are born in your body, so they look like your regular cells. And then if you try and generate treatments to target cancer cells, what you're doing is trying to target cancer angiogenesis or blood vessel growth, or stop the cancer cells from growing too much, or stop the cancer cells from moving into surrounding tissues. But there are other cells in your body that are performing that function all the time. So any of the tumor's abilities that you try and halt, you're going to have side effects by interfering, or side effects that are interfering with normal homeostasis. So it's one of the many reasons why cancer is so difficult to treat. So we will leave that there, and we'll come back and do some more in the next one, the second half of the case study.